Hi Team Pima. Welcome to the second video in a series of 4 videos for the nervous system. This video will talk about membrane potential. Just checking in on my... Okay, so we did this in chapter 9 when we talked about muscle physiology. We went over this review. So this guy right here is a neuron. Here's the axon of the neuron. Oh, this is the cell body of the neuron, and the axon of the neuron is a large process. And then they cut out a piece of the axon, and this yellow line here and here, that's the phospholipid bilayer that surrounds the neuron, including the axon. And we talked about how the inside of the cell is negative relative to the outside of the cell. So these charges line up along the inside of the plasma membrane, these negative charges, and then the positive charges line up along the outside of the plasma membrane of the neuron. Then we talked about how high potassium was higher concentration inside the cell than outside the cell. Sodium is higher concentration outside the cell than inside the cell. Chloride is also higher concentration outside the cell than inside the cell. And then these A minus, those are just large proteins with a negative charge. So all this together gives the inside of the cell a negative charge relative to the outside of the cell. And then we looked at this cartoon over here, and it showed this big arrow shows potassium diffusion, and a smaller arrow shows sodium diffusion. So that just kind of shows that there's more potassium channels in any cell. So at any one time, you could get more potassium diffusing out of the cell down its concentration gradient, because remember, potassium is really high here on the inside of the cell, and then it's lower on the outside of the cell. So that means if a potassium channel is open, then potassium will diffuse down its concentration gradient from the inside of the cell to the outside of the cell. And then this cartoon just showed the sodium-potassium ATPase, and for every 1 ATP that you spend, you can pump sodium against its concentration gradient, 3 sodiums against its concentration gradient to the outside, and 2 potassiums against their concentration gradient to the inside of the cell. So this just tells you a couple bullet points on these cartoons. The negative ICF, or inside the cell, is due to a much greater diffusion of potassium out of the cell. So there's more potassium diffusing out of the cell at any one time. So that, with other things, makes the inside of the cell more negative. And then it says ions, that would be potassium ion, chloride ion, sodium ion. They leak through the membrane and rush in and out through opened gates. So these ions cannot get in or out of the cell unless the gates are open. And then I asked, what maintains a constant state of sodium concentration greater on the outside and potassium on the inside? That would be the sodium-potassium pump. That's this big purple thing here. So if the membrane depolarizes and repolarizes, like during an action potential, the sodium-potassium pump is always there to reestablish the fact that there's more sodium on the outside of the cell than inside the cell, and more potassium on the inside of the cell than outside of the cell. This is a cartoon that shows principles of electricity. So at the bottom part here, when ions, for example, if a potassium channel is open and ions flow, so potassium would flow to the outside, you could get a reading on a voltmeter. So this part of the cartoon, so here's a voltmeter, it's showing minus 70 millivolts. So it looks like it's taking that reading. So here's an electrode taking that reading from the middle of the axon of the neuron. And because there's more negative charges on the inside of the cell than the outside, this resting membrane potential of this axon's neuron reads minus 70 millivolts. So the principles of electricity, opposite charges attract each other. So if you are a positive sodium, you will be attracted to something that's negative, whether it's the inside of the cell or a chloride ion. If you're positive, you'll be attracted to something that's negative. And it says here, energy is required to separate opposite charges across a membrane. So the energy is in the form of ATP with our sodium potassium ATPase. If the opposite charges are separated, then the system has potential energy. So because the plasma membrane here is separating the positive and negative charges, there's potential energy. That means the energy is stored in these ion gradients. And if you open those gates to let the ions flow, then the potential energy can turn to kinetic energy. And then the last bullet says voltage is a measure of potential energy generated by separated charges. So voltage is just a measurement of this potential energy, and you can read that on a voltmeter. So we went over this cartoon in the last video for chapter 11. And this just shows at time at 1 millisecond, voltage-gated sodium channels open in an unmyelinated axon. Voltage-gated sodium channels open in a myelinated axon. And then you can see after 10 milliseconds, look how far the action potential has gotten on a non-myelinated axon. But look how far the action potential has gotten on a myelinated axon. So it just shows that if you are an axon and you are myelinated, you can conduct an action potential with a greater velocity. So I've got here in this cartoon, it says membrane ion channels. There's two types, leak channels and gated channels. And then there's two types of gated channels. So there's ligand gated and voltage gated. And we've talked about these before in chapter 9, but here's a cartoon. It shows a ligand gated, some people call them chemically gated ion channels. So here's our hero, the phospholipid bilayer, and then here's our ligand gated channel, and then there's a receptor here for a neurotransmitter, but there's no neurotransmitter bound there, so it shows the sodium, the yellow sodium ion trying to get in, and it can't because the ligand gated ion channel is closed. Now if you have the neurotransmitter, which is this orange ball, a neurotransmitter binds to the ligand gated ion channel, then the channel opens and sodium, the yellow sodium, can move into the cell down its concentration gradient, and potassium can move out of the cell down its concentration gradient. And no ATP is needed here because these ions, sodium and potassium, are moving with their concentration gradients, not against their concentration gradient. And then this next cartoon, letter B, these are voltage-gated ion channels. So sodium channels kind of, it's really complicated and it's pretty cool actually. So it's a channel, and then it's got a couple different gates. So gates can be closed and capable of opening, and then these gates can be closed and incapable of opening, which we'll see later on. But so in this cartoon, we've got the phospholipid bilayer here. The negative charges are on the inside of the cell. The positive charges are on the outside of the cell. And the voltage-gated channel, see how it looks like it's opened here, but the gates are closed down here. Now the membrane voltage changes. So let's say it goes from minus 70 millivolts to minus 55 millivolts. That's the threshold potential. Now the channel is open, but the gates are also open, so sodium can move into the cell down its concentration gradient. And then, so those are the gated channels. Leak channels, they don't have gates in them, and they're always open. So let's see what we have here. Gated channels, so ligand gated and voltage gated. To open a ligand gated channel, you need a neurotransmitter. To open a voltage gated channel, you need to change the membrane potential or the voltage that that channel is sitting in. And then it doesn't show here, but there's a third type of gated channel called a mechanically gated channel. They respond to mechanical changes, and we'll see mechanically gated channels when we do the special senses. So hearing, seeing, smelling. Okay. So this is just a few more details. It says when the gated channels are open, so here and here, when they're open, ions diffuse down, they diffuse across the membrane down their electrochemical gradients. They move from higher concentration to lower concentration. And then this third bullet or fourth bullet, it says the ion flow, so that's sodium and potassium flowing through the channels, creates an electrical current, and it changes the voltage across the membrane. So for example, it's negative. In this cartoon, it shows it's negative on the inside of the cell. So these negative charges, positive on the outside. But when the voltage-gated sodium channel opens, sodium rushes in. Look what happens to the inside of the membrane. The voltage changes. So now the voltage is positive on the inside and negative on the outside. So this just shows our voltmeter at 70 millivolts and our axon. So generally in neurons, the potential difference across a resting cell, or aka the resting membrane potential, is usually around minus 70 millivolts in neurons. In muscles, it was minus 90. The resting membrane potential is generated by differences in ionic makeup. So sodium is large concentration on the outside, small concentration on the inside, for example. And then also the differential permeability of the plasma membrane. And the permeability is due to channels. So if, for example, all of your sodium channels in a cell were open, then that cell would be very permeable to sodium. And this just gives a few more details on this thing that we call the voltmeter. So that we talked about how this is an electrode. It's actually a microelectrode because you're putting it in a cell. And then you guys that have had electronics, you have to have a ground electrode outside the cell to get the measurement. So we've seen the action potential before when we talked about muscle. Action potential, it says first bullet, is caused by permeability changes in the plasma membrane. So when the membrane becomes permeable to sodium and potassium, then sodium and potassium are going to move in and out of the cell and cause the membrane potential to change. In an action potential, you have a brief reversal of the membrane potential. So in this graph, it was minus 70, and then it reversed. The membrane potential of minus 70 reversed to plus 30. Action potentials and this brief reversal of membrane potential occurs in muscle cells. We saw that in chapter 9, and now we're going to see it in axons of neurons in chapter 11. This action potential will not decrease in magnitude over distance. So this action potential will stay this same strength the whole length of the axon that it's running down. And this is the way the nervous system does long distance communication. So with these action potentials, because the magnitude or the height of this wave doesn't change along the whole length of the axon. So number one, at minus 70, that's the resting membrane potential. That means the voltage-gated sodium and potassium channels aren't closed, have minus 70 millivolts. And then the first phase is called the depolarization phase. We talked about this in muscle physiology. That's where the voltage-gated sodium channels are open in the membrane, and sodium is rushing in, so positive charges are rushing in. That's making the membrane potential, you can say either more positive or less negative, however you like to look at that, whatever makes most sense to you. There's an initial depolarization right here, and if for some reason that depolarization of the resting membrane potential did not reach this thing called threshold potential at -55, so I call threshold potential TP. If this depolarization right here did not reach 55, you won't fire an action potential. But if the depolarization does reach -55, then you fire an action potential, and the action potential is propagated or moves down the length of the axon. So that's a depolarization phase. At the tip of the peak here, remember voltage-gated sodium channels close and voltage-gated potassium channels open. And now during this phase, the repolarization phase, sodium rushes out of the cell down its concentration gradient, and as positive sodium leaves the cell, that means the cell is becoming less positive or more negative. And then you get this little overshoot. So here's resting potential. You get a little overshoot before you come back up to resting membrane potential, and this overshoot is called hyperpolarization. And I said this earlier, this depolarization phase, so this from here to here, this is all the depolarization phase. But if this initial depolarization doesn't reach threshold potential at -55 millivolts, then you don't fire an action potential. If you don't reach -55 millivolts, that's called a subthreshold stimulus, and they call it a local potential. It's not an action potential. If you want to fire an action potential, you are required to reach the threshold potential at -55 millivolts. Once you reach 55, a complete action potential fires. Action potentials they're said to be all or none. So once you reach -55, you will fire an action potential. So this is this is kind of a tricky graph. So it's got, on the x-axis, it's got time in milliseconds, but check it out. On the y-axis, on the right y-axis, it's got our regular millivolts, the membrane potential, but on the other y-axis, it's got relative membrane permeability. So if we're looking at the y-axis, we can see this pink thing, that's our action potential. But now to read sodium and potassium permeability, we have to look at the other y-axis where it says relative membrane permeability. So it's just saying that during the depolarization phase of the action potential, step 2, the membrane is highly permeable to sodium. And then during the repolarization phase, and a little bit here too, a little bit of the depolarization phase, the membrane is permeable to potassium. So propagation of an action potential. We've talked about what an action potential is, what it looks like, what kind of channels it needs, but how do you propagate it? So here's the plasma membrane of an axon, this blue stuff. You can see the inside of the axon is negative here, and it's also negative here. So the inside of the axon is negative both before the stimulus, so this could be a stimulus from an electrode in the lab, but it's negative before and it's negative after the stimulus. But at the stimulus point, that region of the axon is depolarized because sodium is rushing in. So let's look at the bullets. It says a stimulus leads to sodium influx causing a patch of the membrane to depolarize. So here's the stimulus. It's probably an electrode, so they send in some negative charges through this electrode. It's a lab setup. So they stimulate the membrane, sodium flows in, and then it's the second bullet, it says local currents affect adjacent areas in the forward direction. So the current they're talking about is the current, the flow of sodium. So that's a current of sodium flowing in. And So sodium, when it comes in, it's actually going to move this way, and it will move this way too. But the action potential will only go this way. So that's going to be a little tricky, but I'll explain that. So depolarization, so the membrane was polar when it was negative on the inside, positive on the outside. Now it has depolarized when the positive sodium goes in. And then the action potential is going to travel down the length of the axon. So this is a slide that says coding for stimulus intensity. So remember, you can have a stimulus that will cause an action potential to fire. So in this graph, the y-axis is membrane potential, again in millivolts. So there's your resting membrane potential at minus 70 millivolts. Here's plus 30 millivolts. And then these are action potentials firing. They're just showing them as straight lines, not as like the peaks that we see. And then down here, this is set up in a lab. You have the stimulus voltage. So, and it's got 0 millivolts here. And it doesn't have any other millivolts along the y-axis, but that's okay. And then time is on the x-axis for both graphs. So check it out. So they give, in the lab, they can give a stimulus voltage. So here you are at 0, and right here they give a stimulus voltage. But look, it doesn't, that amount of voltage doesn't reach threshold, and you come right up here and you don't see an action potential. The next stimulus they give, check it out, it went past, that stimulus did surpass the threshold stimulus, and you fired 1, 2, 3, 4 action potentials. Now look at this graph. Here the stimulus is bigger than the previous stimulus, and you fired 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11 action potentials within a period of time. So the stronger the stimulus, the higher the frequency of action potentials that you'll fire. So then the bullets say strong stimuli generate action potentials more often than weak stimuli. So this little guy here would be a weak stimulus, this guy would be a strong stimulus, and you get a higher frequency of action potentials with a strong stimulus. Okay, so now let's look at this cartoon. So this is an action potential. So we've got minus 70 millivolts on the y-axis and time in milliseconds on the x-axis. And here's our resting membrane potential along here. And then it shows you, it gives a stimulus right here at 0 milliseconds. And we know the story. Sodium rushes in to the cell, the membrane becomes depolarized, and then at the top, the voltage-gated sodium channel is closed, voltage-gated potassium channels open, and then you get a repolarization event when potassium leaves the cell. But what are these things up here? Absolute refractory period and relative refractory period. So refractory just means that you're refractory to responding to something. So in the absolute refractory period, that's this green section right here. This is absolute refractory period. The sodium gates are closed. So here's a picture. Let me back up. This yellow stuff, that's the plasma membrane. Here's the plasma membrane. Here's the outside of the cell in blue. Here's the inside of the cell, the ICF. During the absolute refractory period of an action potential, these sodium gates are closed and incapable of opening. So sodium can't get in. So you cannot fire another action potential during this time because sodium gates are closed and incapable of opening. That's the absolute refractory period. The relative refractive period, on the other hand, the sodium gates are closed, but they are capable of opening if you fire a strong stimulus. So let's take a look at this, what it says. It says a neuron will not respond to a second stimulus of equal strength to the first stimulus to which it it has already responded because it's in the absolute refractory period. So here's the stimulus. Here's our arrow down here. Here's the stimulus. If you try to hit that neuron with another stimulus, let's say right here in the absolute refractory period, even though that stimulus might be really strong, so that strength of the stimulus is shown by a really strong length of the arrow, you can't fire another action potential during this relative refractory period. However, if you fire a strong stimulus here, or if you give that neuron a strong stimulus here in the relative refractory period, you could fire another action potential. But basically, you have to get a second action potential, you have to have your membrane potential back to or near resting membrane potential before you can fire another action potential. Okay, so we talked about that. I don't know why that's up twice, but it is. So then here's this, here's some bullets and words about the absolute refractory period. Mother Nature created an absolute refractory period because it ensures that each action potential is an all or none event. So you don't get a half-baked action potential. These refractory periods allow each action potential to fire fully. Absolute refractory period also makes sure that you're doing a one-way transmission of nerve impulses, that is from the axon hillock towards the axon terminal. And so that's the absolute refractory period. And then the relative refractory period, if you do fire an exceptionally strong stimulus, you can fire potential. Okay, this is a great cartoon because it shows a lot of detail. So if we start down here at number 1, this is the resting state. So it shows the here's the plasma membrane of the neuron. Here's the outside of the cell in blue, inside of the cell in yellow. It shows your sodium channel. The channel has the gates in it. So in the resting state, the sodium channel, the gates are closed, but they're capable of opening. At resting membrane potential, the potassium channel is closed. And then they show that the sodium channel has gates in it, and this top one is called the activation gate, and the bottom one is the inactivation gate. If it's in this configuration where just one of the gates is closed but the other is open, then that means the sodium channel is closed, but it's capable of opening. So we're here in this part of the graph. Here we're in number 1 when we look at the action potential trace. And then when we come up to number 2 and we get to threshold, that means the activation gate and the inactivation gate, they're both open, so sodium can rush into the cell. Potassium's still closed here. and so sodium will start rushing in and the membrane potential will start to change. That's number 2 right here. Now in this cartoon, threshold potential they have at -50. It doesn't matter, it's -50, -55. But if you reach threshold potential, that means that all your sodium, your voltage-gated sodium channels are open. And that shows here in number 3, all your voltage-gated sodium channels are open, sodium is rushing in. And if we come back over here to the graph, the membrane is getting more and more positive or less negative as sodium rushes in. And then step 4 over here, at the top, remember the voltage-gated sodium channels close and the voltage-gated potassium channels open. And so when the voltage-gated sodium channels close, the inactivation gate closes. And then in this cartoon, it shows that the voltage-gated potassium channels have opened, potassium is leaving the cell. So at this point, in number 4, because the inactivation gate is closed, the activation gate in the sodium channel is still open, but the inactivation gate is closed, that means that channel is closed and incapable of opening. And that's part of the story of the absolute refractory period. You can't fire another action potential during this time because you can't open your voltage-gated sodium channels. Okay, so that's 4. And then you come back here to number 5. You come over here. You get a little undershoot, a little bit negative, a more negative resting membrane potential than you had when you started. And the sodium channels are at this point closed. So both the activation gate and the inactivation gate is closed, but there's still some potassium leaking out, and that's why you get a little bit more negative here at number 5. Okay, conduction and velocity of action potentials. So what allows for an axon to give an action potential a high velocity or a low velocity? So one thing is the effect of the axon diameter. So if you have a really thick diameter of an axon, that means there's more voltage gated sodium potassium channels in that thick diameter axon, so you can increase the velocity of the action potential. So larger diameter fibers also have a decreased resistance to current flow that increases your conduction speed. The second way you can affect the velocity of your action potential is you can myelinate your axon. We showed that. So here's the cartoon of a myelinated axon. Here's the myelin sheath. This looks like it's in the peripheral nervous system. So that would be a Schwann cell myelinating. And then you can see in these nodes, the nodes between the Schwann cell myelination, you see these purple things. Those are all the voltage-gated sodium and potassium channels. So if you are a large diameter axon and you're myelinated, you're going to have the fastest conduction velocity of an impulse, aka an action potential. So what does the myelin sheath do? It insulates the axon, prevents leakage of charges, allows for saltatory conduction so that the action potential could jump from node to jump to the next node. It's called saltatory conduction. So the action potential goes about 30 times faster than an unmyelinated axon. And these voltage-gated sodium and potassium channels are located in the nodes of Ranvier. This just gives you a few more details. Nerve fiber, remember fibers, the other name for fibers in this chapter are axons. So the nerve fiber classification, you can have group A axons. They have a really large diameter and they're myelinated. And they go, you find them in somatic, sensory, and motor fibers. So somatic means the body, so that's sensing things in the muscle or the joints or the skin, that's somatic sensory. And then motor fibers are sending motor neurons with their motor axons out to muscles, skeletal muscles. And this just shows a box that tells you the fiber type. So you could be fiber type A, and within that you could be alpha, beta, I think that's gamma and sigma. And these all function, these are the functions of that fiber type. You have a B fiber, and you find those in postganglionic autonomics, and we'll talk about that when we talk about the autonomic nervous system. And then these C fibers, those are fibers for pain, and they actually travel the slowest. So pain, temperature, and mechanoreception. So group B fibers, I like this. They have an intermediate diameter and they're lightly myelinated. I'd like my fibers lightly myelinated, please. And you find them in the autonomic nervous system. And then group C fibers, we said they have a really small diameter and they're unmyelinated. And you find them in the autonomic nervous system. And we'll talk about these group B and group C fibers when we talk about the autonomic nervous system in a couple of chapters.